Okay, so this lesson in the next few lessons are extremely important and very high yield for the IMAT and BMAT exam. Because these kind of questions are the questions that you are very likely to meet and encounter when it comes to the IMAT and BMAT exams. And they really like to try to set you a trap when it comes to the terminology and this specific topic. So we talked about that during the S phase, we have the DNA replication, in order to allow the cell to prepare for cell division. Because in mitosis, in the future cell duplication, we want to make sure that the future daughter cells will contain the same content, the genetic content. Therefore, we need to duplicate it. Now, we also talked about the fact that chromosomes are the most compact way to actually compact the DNA and the genetic material before we actually proceed to the replication. Because if you think about it, before the preparation for the actual mitotic phase, the M phase of the cell cycle, the replication, the duplication of the cell, the DNA is open in a chromatin, and it's not dense because we need to translate and transcribe different proteins and RNA from this DNA, so we can't make it dense. Therefore, you have a lot of molecules just floating around inside your nucleus. The compact way to actually take the following molecules is the chromosomes. So they can come and they can become very dense structures. Now humans, and from this point it's very important, humans have 46 chromosomes. We have 46 chromosomes, but we actually have 23 chromosomes in pairs. So we have 23 pairs of chromosomes. We have 23 pairs of chromosome because one comes from the mother, which is the maternal, and one comes from the father, which is the paternal. Because we have the maternal and the paternal genetic material when it comes to the chromosomes, we call our genetic material in our animal cells, we call it diploid. Di for two. So we have one maternal, one paternal. So I want to show you a picture of something called the karyotype. Karyotype is when you take the chromosomes and you map them down in the lab in order to see their different sizes and length, and if everything is correct and you don't have any duplication of different chromosomes. So it's a common way to actually check the chromosomes. So right now you see the karyotype over here. And as you can see, we have 23 pairs. Just for your general knowledge, we have 22 pairs that are called the autosomes. And we have the XY, which are the sex chromosomes. They are not actually a pair because they are different. When it comes to a woman, you will have XX, then it will actually be a pair. But when it comes to a male, you will have XY. Just for your general knowledge, don't worry, we will talk about it in the future. For now, remember that we have 23 pairs. And each one of these pairs is a DNA molecule. So this is, let's say, is the maternal. And this is the paternal DNA. Now, these DNA molecules, each one is a single DNA molecule. So chromosome 1 in dipole cells will have 2 DNA molecules. One from the father, one from the mother. The chromosome 2 will have 2 as well. Chromosome 3 will have 2 as well. So on and so on. Now, this is before the actual DNA replication. Before the DNA replication, each chromosome will contain one DNA molecule, and each molecule will contain genes. You will see that for each gene, we have 2 alleles. alleles are an abbreviation, are a different form of the same gene. For example, I have a gene for eye color. This gene is found on chromosome 1, just an example, on chromosome 1, on nucleotides 300 all the way to 700. Just an example. The same location on both DNA molecules will be called the specific gene for the eye color. However, if the father or the mother will have a different form of the same gene, different sequence, for example, the father will code for blue eyes and the mother for brown eyes, we will have different alleles. So between the 300 nucleotide and the 700, on both DNA molecules, they code for the same gene, but they are different alleles. It's the same gene. It's the blue eye, it's the eye color gene, but the allele itself is different. It varies between the different kind of alleles. So now we understood two things. The DNA molecules between the father and the mother can vary, and the DNA molecules are one for the mother and one for the father. Because we have two DNA molecules, we have two alleles for one gene, we are going to have a dipole cell. And humans are dipole organisms. What happens after we are replicating the DNA? So I have another karyotype over here. And this time we have 46 pairs of chromosomes. So we have 1, 2 times 46. 23 in this case. Times 23. So 2 times 23 will give us 46 pairs of chromosomes. However, you will see that these molecules and this molecule, both of them are called one chromosome. And in this karyotype as well, both of them are called one chromosome. What defines chromosome is something called a centromere. And a centromere is a protein that attaches the two, you will see, sister chromatids together. So chromatid, we said, is a compact and way to form the DNA around the proteins. When you go from this to the compact way to actually form the chromosomes, you will have chromatids and chromosomes. And the chromatid, in the middle, you will see something called a centromere. Now, this one is called a chromatid, and this one is also called a chromatid. They are different chromatids from each other because one is maternal and the other is paternal. However, when it comes to the karyotype that occurs after the duplication, after the cell, you will have this one is paternal, but this one is paternal as well. And this one is maternal, but this one is maternal as well. Which means that you have two chromatids that are basically the duplication of each other. Therefore, we call them sister chromatids. And once you learn in the future about the chromosomes, you will see that between the sister chromatids, you have something called a centromere. Let's choose another color. Centromere. A nice trick, and remember it for the IMAT and BMAT exams, will be to count the number of centromeres to see how many chromosomes we have, because the number of centromeres will show us how many chromosomes we have. It directly associates to the number of chromosomes. You will see it in the future. But in this video, I would like to show you and to focus on the number of DNA molecules. So now we have 1 pair and 2 pair. This pair, each one is going to contain 2 DNA molecules. 2 for the mother and 2 for the father. Let's take another color to differentiate between the paternal and maternal DNA molecules. So now in total we have 4 DNA molecules for each area that is called a chromosome basically. So we said we have 23 kinds of chromosomes. In our case we have 24 because we have also the Y. Therefore you're going to have 1, 2, 3, 4 times 22, which is going to give us 88. And 1, 2, 3, 4 plus 4. You're going to have 92 DNA molecules after the replication and before the separation of the daughter cells when it comes to the mitosis phase, which we are going to study. But you are going to have 1, 2, 3, 4. You're going to have 46 chromosomes because you have 46 centromeres that are going to hold everything together. The sister chromatids, because they are still yet to separate during our mitotic phase, they are still called a single chromosome. But this time, after the replication, we have a single chromosome that is composed from 2 DNA molecules, not a single one like we saw over here. So over here, we still have 46 chromosomes because we still have 46 centrosomes. However, here, each chromosome is composed out of a single DNA molecule. 1 for the maternal and 1 for the paternal. But when it comes after the replication, and I know I'm repeating it a lot, but it's so important to remember for the exam, we still have 46 chromosomes, but we have 2 DNA molecules for 1 chromosome. 2 for the maternal and 2 for the paternal. And this is a very important point to remember, which you are going to see in the future. Now, in the next few lessons, we are going to actually understand how the cell replicates and separate. You are going to see that you have the microtubules coming and attaching to this area close to the centromere. And the centromeres, this one and this one, they are going to go to the separate sides. But you will see that once you take each one, because you want one paternal to one cell and the same paternal duplication to another cell, because you want to have the same exactly genetic material, once these two are separated from each other, you will see that they have 2 new centromeres instead of only 1. Therefore, once the broken down and the separation of the DNA molecules actually occurs in order to migrate them to the other poles of the cell during mitosis, then you will have double the amount of chromosomes, only after the separation. I hope this video made sense. I know it's a bit heavy. We are going to break it down lesson by lesson in the future. But right now, remember what I said in this video. Remember the amount of DNA molecules you have in each state before the replication and after the replication. Remember what a chromosome is, and that the number of chromosomes inside your cell is directly correlated to the number of centromeres you have. And you will see everything in the next few lessons.